So the main thing about cell structure and function is that eukaryotic cells have these membrane-bound compartments known as organelles. Different organelles have different specialized roles within the cell, but mainly the purpose of everybody together is to maintain homeostasis and to pass on genetic information to your offspring. So the different organelles have really, really specific things that they're each in charge of. And of course, we're going to go through them individually and very, very specifically in terms of how much the AP exam wants to know in terms of the level of depth regarding each of these organelles. Cellular energetics. So we talked about this just a little bit already. As a brief reminder, ATP is the currency that the cell needs to do reactions that are what are considered not favorable. So if a reaction is not favorable, it's not going to happen. However, if you now add ATP to that reaction, it's more likely to happen. And we'll go over thermodynamically why that's energetically more likely to happen. So cellular energetics is basically talking about using the chemical principles that we know to now see if certain reactions are going to happen in our bodies and to see why if certain reactions that aren't going to happen and we still need to happen, how our body uses ATP as a vital currency to make sure that that happens. So that's all about energy regulation in the cell. Cell communication and the cell cycle. So for example, if there is a pathogen that attacks one of our cells have certain messengers and certain cascades that are going to get activated to trigger their buddies and say, hey, there's a problem here. Let's keep triggering people until we automatically trigger a protein that's going to come and destroy this cell so as not to spread this infection to the rest of the body. This is just one example of the many, many different aspects of cell communication that we're going to be going through. A cell also needs to grow individually in its own lifetime. This is called the cell cycle. So the cell cycle is made up of two core phases. One of them is called interphase, which is basically a growth and regulation phase. And the second phase is called mitosis. So what happens during interphase is that the vast majority of the cells spend its lifetime in interphase. That's 75% of the cell cycle is spent in interphase. Basically here we're going to be growing and we're going to be replicating our DNA. Now it's during mitosis that we actually get one cell splitting to become two cells. This is how growth happens, right? So the difference between a baby and an adult from a purely physical point of view is basically many series of mitosis, okay? So that's a little bit about the cell cycle. So now on to unit 5, heredity. So the DNA that we're going to be learning about during the chemistry of life and a little bit during the cell cycle is, of course, the heritable material in a eukaryotic and a prokaryotic system. So cells need to have a way to pass on the information that they have used to success, otherwise known as their genetic information, to their offspring. This is one of the tenets, this is what's considered one of the tenets of life, to pass on your information, your genetic information to your offspring. There are many different patterns and many different more intricacies in terms of how this exactly happens. And that's what we'll be going through in heredity. But heredity kind of also leads into gene expression as well, because not every single gene needs to be on all the time. If you have a certain gene that activates a protein product that you need to metabolize sugar and you're not eating sugar, you don't really need that gene to be active right now. So that's an example of the importance of gene expression. In the same context, regulation, right? So like I said, when you're not eating sugar, you're not going to have the gene that encodes for the protein that does regulation because you don't need that. And like I said, your body wants to maintain, your cells want to maintain a constant homeostasis without expending too much extra energy where it's not needed. So why make the protein for no reason? Let's turn off that gene and use our precious ATP elsewhere. Natural selection I already talked about a bit. So mutations are random. Some of them are beneficial. The beneficial ones are what get passed down from generation to generation. And over a period of time, populations evolve. That's the theory behind natural selection. Ecology is a little bit more of a macro approach. So again, through these units 1 through 7, we learn about organisms, we learn about organelles. That's more of a micro approach. So now we're seeing how these organisms interact within different systems. Mostly the AP exam focuses on how non-human organisms interact from an ecological point of view. So we're going to be learning about different groups of animals, how they're competitors in certain biomes. Biome is basically like a, short term is just like a weather condition, like a massive region where the weather condition is more or less similar in that region. So you call that entire region a biome. You're basically going to be learning about how different organisms interact within the biome as well. All right. So that's a little bit about ecology. Yeah, guys, please feel free to ask questions. I see the chat's pretty empty. If you guys have questions about anything that I'm saying, please feel free to let me know. Sure. Yes. Right. Yes, I am at a college. Yes. Yes. That is why I have done this. Sure. So the vast majority of what's on the AP exam is actually going to be directly relevant to most of the undergraduate level college courses that you're going to be taking. I have students that I teach right now that when I ask them about their APs, I see the students that are succeeded in the college courses that I teach now. I see that they already have a pretty good background from their AP courses. Students that I see that struggle a little more either didn't take the AP course or took the AP course but didn't take the exam too seriously. They didn't really study for it, and they're a little behind everybody else right now. So what are the courses that I teach? I teach molecular biology, which plays very, very heavily into the chemistry of life and cell structure and function. I teach biochemistry, which plays a lot on cell energetics. I used to teach introductory biology, which is basically kind of like an ecology class where we learn about natural selection too. So all of these courses throughout one point or another, I have had experience teaching the topics covered on the AP exam at the college level. So when I came to Francis, this is why I thought that this would be the most optimal course for me to teach, given that I have so much experience at the undergraduate level about what all of these different topics are going to be like for you guys when you get to college, assuming, of course, that you do continue on the pre-bio field, which I would recommend. Yeah, exactly. Likewise. Yes, definitely. Definitely. Exactly. This is basically what I'm looking at now. These topics are the starting point of all biology. I mean, especially units 1 to 6. You guys will be seeing this again throughout. I'm going to say all of your college biology courses. Unit 1, you may even see in your Gen Chem course that will help you there as well. So again, like I said before, I would use this opportunity when we're studying for this AP exam, especially since I personally have experience teaching undergraduate level courses, to really not just get a 5 on your AP exam, but to also make sure that you're set for your next 4 years of undergraduate level bio that you're going to be taking. In terms of standardized exams, I know that some of you are potentially pre-pharmacy, pre-med, pre-dental. This information is also very, very heavily covered on those standardized exams as well. And when the time comes for that, hopefully Kweller Prep does have options for that prep. But again, this is just kind of an introduction to those options. Sure, I do. Right, definitely. So these are probably some of the more highest yield MCAT topics that you can learn at the high school level even. Like I said in the beginning, just in case anybody missed it, I did score in the 96th percentile on this section on my MCAT. So obviously I was able to get a pretty solid grasp of what's tested on the exam. I've been tutoring MCAT personally for a little bit under a year, so I also know how to explain the relevant information that's going to be on the exam, and that's basically everything that we're seeing right now. So again, getting a great understanding of this right now is basically going to massively assist your career development over the course of the next 5 years directly. And then of course your MCAT score, your DAT score, your PCAT score is going to help you get into grad school. So indirectly, this can also help you get into graduate schools as well. And you guys are at the high school level right now, so this is insane. Sure. So basically, if I had a Venn diagram in terms of the material that was going to be on the AP bio exam and the material that's going to be on the SAT2 bio exam, I basically wouldn't have a Venn diagram. I would just have a very, very big circle. That speaks to the amount of overlap that College Board puts between the AP and between the SAT2 bio. It's the same company that makes these questions. It's the same group of doctors and PhDs that they have that work through all these questions together. And let me tell you, many of them are pretty lazy. So they will have a lot of question overlap between the SAT2 bio and between the AP bio. Specifically, the SAT2 bio is a little bit more focused towards the molecular and cellular aspect of things. And I know this from not only my personal studying with the SAT2 bio course, but through helping different students with that course as well. And the content overlap between the AP bio and the SAT bio is pretty insane. I mean, of course, there are certain strategies to really excel on the SAT2 bio that require separate time, not thinking about APs, but just focusing on SAT2 bio. And of course, there are certain strategies that work for AP, but not vice versa. But in terms of the content and the base material that's going to be covered, which is what we're going to be going through first before we get into the more advanced exam prep, I can definitely say that there's a lot of overlap, if not pure overlap, between the SAT2 bio and the AP bio exams. Right. Sure. So basically, for those of you that don't know yet, most prestigious undergraduate level schools and less prestigious schools that have competitive scholarships are going to be looking at your SAT2 scores. So if I were you guys, and I'm already going to be studying a very similar content set for my AP bio exam, I would not wait until I need to know this exam again during the next year for my SAT2 course, like Francis said, but I would knock out two birds with one stone, basically. You're almost taking the same exam with very little difference in the middle. Of course, there's some difference. There are different exams. College Board does have a little bit of a reputation to keep up, so they made the exams a little different. Sure, but it's basically that you don't want to relearn what it is that you're going to be learning now a year later when you're already going to have to take your SAT2 bio course and your SAT2 bio exam for your top undergrad school anyway. So that's just a waste of time. Otherwise, you can knock out two birds with one stone, and that's just the most efficient use of your time in high school. right? Sure. So tonicity, oh, sorry, I think I accidentally turned off my video. Tonicity and osmoregulation. So tonicity is basically the measuring of salt within a system. Osmoregulation is when you have too much salt in one area. Water is going to literally come to equalize the amount of salt that you have in that area. Then you have a bigger volume because the water came, right? So osmoregulation is all about how water can move across certain membranes to equalize your salt concentration. And again, this is to balance that homeostasis theme that we were talking about before. So the AP also likes to test you guys on thematic concepts as well. How well do you understand a big, kind of macro biology concepts? One of these big concepts is homeostasis. So osmoregulation, tonicity regulation, this is basically how water has the chemical ability. So again, like I said before, biology is difficult chemistry. So water has the chemical ability to equalize out a salt concentration by moving across a membrane. Okay, and that's what osmoregulation is. All right, so water is regulating tonicity, which is salt concentration. All right, sure. Right. Sure. So compartmentalization. If you have a government or society or any big bureaucracy, you want people that are good at certain roles placed in those roles. That's basically why we have organelles. So organelles are made up chemically and physically in such a way that they're specialized and they've been designed that way to fulfill their role within that cell. So for example, there's an organelle known as a ribosome. This is called compartmentalization because the ribosome is physically sealed off. It has a membrane around it, so the ribosome just kind of looks like a circle. And that ribosome has the proteins that are special to make more proteins. So the ribosome is how you make more proteins within a cell. There's another organelle known as the nucleus that I touched about before. The nucleus is kind of like the president of the cell. You have all of the DNA in your cell is stored in the nucleus, at least the vast majority of it. So in the nucleus, you have two membranes. You guys can kind of think of that as like two bodyguards because the nucleus is very important. So you have two membranes that compartmentalize the nucleus. And the reason that you want to do this is because there are certain chemical interactions that are happening in your cell outside of your nucleus, outside of your ribosome, outside of your other organelles, that you don't want interfering with the very specific and very controlled reactions that you have happening within these organelles. All right, so that's why we have different compartments within our cells that are kind of bounded by membranes to help each organelle fulfill its specific purpose and as such contribute to general homeostasis of the cell. All right. Cell energetics. So this gets into a lot of kind of biochemistry topics. So biochemistry is formally considered the study of biology with a chemical background. So it's not chemistry with biology, it's the study of biology with a chemical background. Like I said before, energetics are very heavily tested on your AP. So you have a currency known as ATP, and basically how you build up that molecule and how you break down that molecule is the fundamental understanding of your metabolic processes in that cell. There are certain metabolic processes that you activate when you're low on energy. There are certain metabolic processes that you activate when you're high on energy, et cetera, et cetera. So it's all about regulating. It's all about up, down. And throughout the course, we'll learn about these different pathways. Some of the main ones being glycolysis, the citric acid cycle, the electron transport chain, and then the reverse of glycolysis, which is known as glycogenolysis. So we'll go through all of those. At the same time, Going back to cellular energetics a little bit, we also have these kind of units in our body that are special for doing stuff. Why are they special for doing stuff? Because they have very interesting chemical properties. These chemical properties are held in what are called amino acids. I'm sure you guys have heard the term amino acids before. So amino acids are basically strings of different molecules, compounds, that when you string them up, and the ribosome strings them up, when you string them up, you make a protein. Protein and enzyme, for our purposes, are more or less the same idea. So what this enzyme does is that it binds to a molecule and it makes it another molecule, right? And that's pretty cool. That's how we have a lot of the different metabolic reactions within our cells happen. Enzyme is going to take one molecule and through a process of bioenergetics that we're going to get through more deeply in the course, convert it into a product molecule. It's the main idea behind this topic. So I think we can move on to the next one if we still have time left. I think we're good for maybe one or two more topics. Enzyme structure, sure. So the enzyme is structured in a way that it has a special binding place to that molecule that it wants to change. The enzyme structure has a place known as an active site. The active site is the place on the enzyme where a substrate binds to then become a product. And the substrate and the product have fundamentally different structures that you would not have been able to. Sorry. Yeah, no worries. So enzyme structure, enzyme has a place called the active site. The active site binds the active site binds a substrate to create it into a product. The substrate and the product would not have been able to convert between one to another without an enzyme in a biological context. All right. So this is the importance of these things called enzymes, and we're definitely going to get in a lot more to enzymes. Enzymes are very, very important for your AP, so we're going to talk a lot more about them throughout the course. Now, unfortunately, we have certain environmental factors that can damage these enzymes. These are very, very fragile things. So we're also going to go through how your acidity, your basicity, and your temperature can affect the structure and stability of the enzyme. Cellular energy. So, like I said before, the energy currency of the cell is ATP, and you have different metabolic reactions that regulate the amount of ATP that you produce, where you produce it, when you produce it, what cell is producing more today, what cell is not producing as much today, et cetera, et cetera. These are vastly intricate steps of biological metabolic processes that we're going to get to throughout the course. All right, so I think we're good on cellular energy for now. It's kind of a very broad overview. Photosynthesis. So photosynthesis is basically a broad overview of the way that the plant cell makes ATP for itself. Okay, so as we all know the formula, light, carbon dioxide and water produce as products oxygen, glucose, and some other stuff for the cell. So the plant cell needs the glucose to make it into ATP, and we need the oxygen. This is also an example of a relationship between two organisms. So something like photosynthesis can be tested not only in the context of general knowledge of photosynthesis, but also in a more Unit 8 context of an ecological overview. All right. Cellular respiration is basically how you get certain molecules known as NADH and FADH2, and how through a complex process that would maybe be a little bit difficult to give an overview for today, through a complex process, you generate ATP throughout the use of these molecules, NADH and FADH2. These molecules are called cofactors, and we'll get to what cofactors are and why they're called cofactors when we do cover this throughout the middle part of our course. All right, fitness. No, this does not mean going to the gym. Gyms are closed now anyway. Biological fitness refers to an ability of an organism to pass down its genetic information to its offspring, and that's really the main idea behind fitness. So cells need to talk to each other. If there's a viral cell that's invading, right, and the cell gets damaged by that viral cell, that cell that got damaged needs to give its cell's friends, like, watch out, there's a virus. Please, can we have a protein or a cell come in to destroy the virus? So cell communication is very important, as is the cell cycle. If we can just scroll down a little bit more to see the exact specifics about cell communication that we can go over for the purposes of the AP at least. All right, cool. So signal transduction. What is signal transduction and what are signal transduction pathways? It's basically like a relay race, right? So if you have 10 runners, right, they're each going to be handing a baton to each other. The baton is going to be transferred 10 times. Each of those 10 times, for our purposes, is a reaction. But ultimately, it's the first runner that indicates all of this, and it's eventually going to get to the last runner. So that's how signal transduction works. You're going to keep pinging and ponding until you get to the final recipient of your message. All right. Cell cycle, like I said, you have two main phases: interphase and mitosis. During the interphase, you're going to be growing and you're going to be making DNA. And during the mitosis is when one cell is going to become two cells, and that's how growth happens. So I think we're good with this section as well. A little more specifically about cell communication here. Signal transduction, we chatted about a little bit, but again, just as an overview, as a broad intro, think of the relay race. Think of one person handing a baton to the next person with the ultimate goal of getting it to the final guy, and that's the main thing with signal transduction. So feedback loops are actually very interesting. So if I do not want a certain amount of blood or a certain amount of sugar in my blood, I'm going to have a feedback loop for when my sugar levels get too high. So let's say I have reactions 1 to 10. Reactions 1 to 10 start out with product A and convert it to product B. Okay, so now product B, you may not want to get product B too high. So what you're going to do is you're going to go back and have high levels of product B serve as indicators for previous steps of your pathway to get the product B get inhibited. That's an example of negative feedback. There's also positive feedback, which we're going to go through throughout the course as well, but negative is a little bit easier to understand. Cell cycle I kind of talked about already in terms of the interphase and the mitosis phases, but the cell cycle needs to be meticulously regulated. So when you're copying your DNA and when you're making new cells, you make a very, 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 very dangerous situation for cancer to happen, because cancer happens during DNA mutations, which are going to happen when you're replicating your DNA, and then cancer also needs to spread, and this is going to happen when one cells become two cells during mitosis. So there's a very important regulation of the cell cycle by a protein known as p53, which we're going to get to when we do the actual course, but that's very, very important, otherwise most of us wouldn't be here. Heredity. So like I said before, biological fitness, evolutionary fitness is how well can you pass on your genes to your offspring, right? So that's kind of the very, very, very broad overview of what this unit is going to be. I'm not sure how many of you guys have heard of Charles Mendel. So Charles Mendel is basically the father of genetics, and he had certain experiments that basically produced what we now understand to be Mendelian genetics. Mendelian genetics is probably one of the highest yield concepts that you can test on AP biology. Basically, there are different formulas and different diagrams that you need to understand that Mendelian genetics produced, and we're going to be going over how to interpret, understand, and go through each one of these types of Mendelian diagrams. Okay, so that's the main thing about heredity. I think we're running a little bit low on time. Yeah. Right. Of course. Yeah. No problem. No problem, guys. Thank you. Likewise. Yeah. Sure. So the AP Bio and AP Chem books that I'm working on are basically going to cover the most important material that I feel is on these exams. A lot of times with these companies like Kaplan and Princeton, they give you so much blubber that you don't need to actually do well in the exam, and they don't really do a good job of highlighting what you need to do well in the exam, which is of course the important stuff. So I came to Francis with this idea, like let's reduce the amount of filler that these books have and just gives these kids what they need for the exam to get fives, not even fours, fives. That's going to be the purpose of this book, to get a five. So it's going to be based in content on the overview that we just went through in terms of units 1 through 8. And then I'm going to be combining the different mnemonics and study techniques that I picked up throughout my own MCAT studying and my own teaching of undergraduate courses to go through what the best way is to present this not so easy but very, very fun information that the AP Bio exam is really focused on in these books. These books are going to have practice questions, and each book is going to be a few hundred pages. So even though we're being efficient and we're not giving you guys filler and blubber, we're also giving you guys enough material and a good amount of content to get a file on this exam. Yeah, likewise. Can't wait. No problem.